high, love, raising high, love.
My talk today is Biology is Changing Everything. Thank you. And, I think that, in 20, 50 years from now, people are going to look back on today as the early years of what will probably be called the century of biology. And you might be wondering, what was the 20th century? That was really the century of physics that brought us computers and internet and all of those things. But really poised for the future is biology. And so that's going to be our focus. And what I thought I'd do is just start off a bit to give you an intro of kind of who I am in becoming a scientist and why. And then we'll talk about some of the key areas of biology, and in particular, this area called synthetic biology, which is playing a really big role and has, I think, the potential to be a very new paradigm for addressing some of the biggest challenges in the world today, like energy and healthcare. And synthetic biology is really enabled by our ability to read and write DNA, so we'll talk a bit about that, and then talk about what folks are really doing with it today and maybe tomorrow in the future. So when I was like 4 or 5, I was very much like this little girl and really interested in how things worked. I especially liked math and things that were analytical. And I also really liked science in terms of understanding how things do work and why things come together or not and react. And, growing up, I was really interested in both math and science. And one of the questions that I often get is whether or not, as a young girl, there was ever, this feeling of, gender bias of not, that not being the right thing to do. But in fact, my parents and friends always said that I could do, anything. And there was only one time that I really experienced any real gender bias. And that was in middle school. And we were all allowed to sign up for 2 electives. And so I signed up for French because I was continuing to take French as a language, and electronics. And when I got my card back with my classes on it, in fact, I was told that I wasn't going to be in electronics, but I was going to be in typing. And I was kind of ticked off. And it's kind of interesting because in the, in our digital keyboard world of today, I can't really complain. I mean, it's good to know how to type, right? But I'm also the head of research for the world's largest electronics company. And it wouldn't have been bad to have been able to start my electronics interests a little bit sooner too. So anyway, that was really the only time. And in general, I've had incredible opportunities to go off in science and math. When I was in high school, I thought I was going to be a math major and took lots of math classes, really enjoyed math. And I did my undergrad at Stanford, and my first year stayed as a math major. But over time, I started thinking more about what my career would be and what I would do. And as I looked at math, I saw it more narrowly than I probably should have. But in fact, I thought, I wanted to maybe take a change. And so I took a whole bunch of science classes my next couple of years, lots of chemistry, biology, and physics, and decided that I really liked chemistry. I really liked understanding why gemstones were all different colors, or how we got energy from the food that we eat, or how our immune system keeps us healthy, things like that. And so with that, I ended up staying with chemistry, majoring in chemistry, and then doing my PhD work also in chemistry, in bioinorganic chemistry at MIT. And then I went to work in industry. And this is actually the first picture that really is me. And that was in my lab at Hewlett Packard Laboratories. I worked for HP Research when I first started my career. And you can see I was working with a lot of electronics equipment and chemicals as we were trying to understand measurements inside of blood and stuff. And I worked as a scientist for several years, and then had an opportunity to move into management. And I enjoyed management a lot. And then HP spun out a new company, and that was Agilent, which is where I work today. And in management, I found that I could really think about lots of different things, and I liked that. and I also liked working more with people and helping remove obstacles and stuff. So I continued in management and, continued to grow in the role. And so I've been now leading research for all of Agilent for a little over 10 years now. So synthetic biology is all about using genes, which are DNA, in order to program cells to become cellular factories that can then produce high-value products. Today, in the world, we have a lot of challenges as, growing populations and climate change and developing economies really create dilemmas and trade-offs as we try to nurture the health of the world's people and our environment and our cities. And with that, it's really time to think about, what is the world going to be like in 20 or 50 years? And I think that in applying biology and some of the things we're going to talk about this afternoon, you can really see how we might be able to meet those challenges in a much better and much more sustainable kind of way. Synthetic biology is the engineering of biology. Biology has changed a whole bunch in the last 20 years, and you don't quite realize that when you look at a textbook or something. But biology used to be a very qualitative science, very descriptive. And in the last 20 years, it's become much more quantitative and measurable. And that's a lot of what my work has been about. And synthetic biology is kind of taking that to the next level, not just being quantitative, but really being able to model and predict biology, much like we do physics or engineering today. You can build a bridge, you can design a circuit, and you usually know that it's going to work. Biology is not like that. There's lots of stuff still to be understood, but that's where people are trying to go. And so in the past, we talked about oil-based feedstocks like petroleum and natural gas being the basis for industrial processes through chemistry that led to different products. And what we're talking about here is bio-based feedstocks. So now we're talking about microorganisms and bacteria and plants like algae actually being fed different materials like sugars or starch or plants, and then converting those into new chemicals and biochemicals that in fact lead to those same types of products. And so we're going to give some examples of that so that hopefully that will make more sense. So with synthetic biology, we're looking at these cellular organisms. And so when we think about producing products, we're really thinking about factories. So they're going to look a lot more like these algae fields or various racks of plants compared to industry and steel tanks and things like that we think about today. The kinds of products that we can re-engineer organisms to produce are really far-ranging, and some of them are illustrated here. It goes from medicines to chemicals and energy, and really being able to make things like car tires and things like that based on biological-based manufacturing. But engineering cells to make things is actually really difficult, and that's because we don't really understand biology as well as we need to be able to go in there and say, I'm going to reprogram this algae or this E. coli to do something it wasn't naturally meant to do. But if you think about it, there's a lot of biology inside those organisms that produces things like natural products, and lots of special drugs are isolated from plants and rainforests and things like that. So in a sense, we're just taking that same biology and just tweaking it in different ways. Biology is well understood, but there's still a long way to go. And so this is just an illustration of how complicated even a simple reaction is. So when we think about this mouse and moving his leg, that's based on this very simple chemical reaction here. It's a metabolite creatine, which is going to creatine phosphate with an enzyme, which is this creatine kinase, and some energy with molecules that provide energy biologically. And that's a pretty simple reaction. But there's all this other stuff going on. And in fact, there's even more stuff. There's this whole network of biological pathways. And in fact, that's what's understood today actually pretty well. This isn't all the stuff we don't understand. And this complex biology or biochemistry is actually going on inside of every one of us, inside all of our cells every day, with all the different functions that let us be, who we are as, healthy thinking people. So when we think about biology and being able to go in and alter some of these pathways, it's easy to do something you think you're doing, but then you might see something that you didn't predict because there is so much complexity there. A lot of what has really accelerated biology is being able to read and write DNA. So DNA is in our chromosomes, and we have this double helix of the A's and T's and G's and C's pairing with each other. And when we look at DNA, there's lots of different things that DNA does. And we can have DNA coding for proteins through genes, so we have DNA to RNA to proteins. There's also a lot of other things DNA does that have nothing to do with genes, which are functional RNAs and non-coding RNAs and regulatory and things like that. But it's really the fact that DNA is such a special molecule that really allows us to do some of the things we're going to talk about. There's some kind of interesting facts about DNA, and so I'm going to just ask, so the DNA inside of our cells is actually coiled up very, very tightly. But if you were to stretch that DNA out, anybody have any guesses as to how long? I think it was,
like calculated in miles?
so it's not quite miles, but we're going to get to miles. If you take all of the DNA inside the human body, absolutely. If you look at just one cell, one more, I think another sign here. It's less than miles. It's in feet, okay? Yeah, so it's a couple of meters, so it's 6 feet. That 6 feet of DNA, when it's tightly coiled up, fits inside the volume of the nucleus of the cell, and that nucleus is only 100 cubic microns, which is almost remarkable, right? I mean, how does it possibly naturally find itself in that really tight way and sit there just ready to do what it needs to do the next time the cell divides? So now, as we think more about the whole human, so there's one cell has that much DNA. Any idea how many cells are inside each of us? Sure.
10 trillion.
Oh my goodness. Did somebody say, wow, look at that. That was a great guess. So we have millions, billions, trillions. That's a ton of cells. And so now, if you take the DNA in those 10 trillion cells and stretch them end to end, you'll go to the sun and back 30 times inside each of us. So remarkable. The piece that actually has motivated a lot of my research and the work that we're doing in RNA at Agilent is the fact that when you look at organisms, we all have genes. And if you look at a worm, a mouse, and a person, we all have about 20,000 protein coding genes. And that always really bothered me because, we are very different than worms and mice. And so there must be a lot more to biology than just the coding genes. And understanding some of the other things that go on inside of cells is a lot of what's kind of drawn my work. But it is pretty amazing that at a DNA level, we're actually not that different than the worms or the mice. So DNA is pretty cool. And what is so powerful is that our DNA stores all of our biological information. It's kind of like the software inside of us. And so this information is encoded by the specific sequence of those A's, C's, T's, and G's that could stretch out between here and the sun all those times. And so this digital storage of biological information is what enables synthetic biology. The genetic code has DNA translating into proteins via RNA. And depending upon how the different blocks of sequence are assembled, you program the cell to produce different amino acids and different proteins. And because the genetic code is conserved across all of life, meaning not just the mice and the worms in us, but also plants and bacteria, viruses are all based on DNA, it means that we can actually move genetic material across different organisms and reprogram organisms, which is what synthetic biology is all about. And to be able to do this editing or changing of genomes across organisms, the easiest way to do that is by varying the DNA sequence inside of the cell. And so sequencing DNA is something that has really accelerated in the last few years. It's what we call reading DNA is sequencing DNA, and writing DNA is synthesizing it. And sequencing has become much easier and much faster and lower cost, but synthesizing DNA is still a lot harder and a lot more expensive. And that is a lot of, again, what my research group does at Agilent is makes very high quality, very long DNA synthetically. And the idea is if we can reduce the cost of DNA synthesis and make it easier to assemble genes, then it's easier to go in and reprogram bacteria to do things that can be very valuable in terms of producing things that we as people really appreciate, like energy and fabrics and things like that. This is actually our manufacturing facility in Santa Clara. I work in California. And in this area, we do lots of printing or writing of DNA on glass substrates. And then we can use that DNA either on the surface or release it in order to do other types of biochemistry. So the DNA printing is on these large sheets of glass, and we actually use inkjet printing. Have you guys had inkjet printers or in school or at home, right? So normally the ink cartridges have ink in them, so we put the bases A, G, T, and C inside the ink cartridges so that you actually print bases instead. One of the biggest contributions was from a fellow by the name of Jay Kiesling. He's a professor at Berkeley, and he figured out how to use yeast to produce an anti-malarial drug called artemisinin. And this is something that had been isolated from sweet wood roots of a plant, but it was very difficult to get. And now through synthetic biology, we can actually produce this drug. In fact, 34 tons are being produced this year through Sanofi using yeast to be able to make this available as an anti-malarial throughout the world. And there's lots of places in our homes where synthetic biology has products today, whether it's fabrics or plastic bottles or contact lens solutions or anti-aging products, all kinds of stuff are based on synthetic biology. So I believe that as we look out to the future, there's lots of opportunities, and even today, there's folks that are working on growing meat in test tubes, figuring out ways to better fight infectious diseases with new vaccines based on synthetic biology, bacteria that can help destroy environmental pollutants, and maybe someday being able to make energy from carbon dioxide and sunlight by reprogramming algae. There's lots of ways to get involved, and iGEM, if you're interested, you can check on the web, is a great place for high school students to form teams to work on some of these kinds of areas. So I really appreciate all of you joining us, and maybe some of you can help change our world and make it a better place through biology. Thank you.